So, uh, very good afternoon, class. How are you all? Please mute yourself and type everything in the chatting. Your doubts or any questions you have while we are having it. So at the moment, let the students join in. So far, 33 students have been added. Let more students add in. Um, then we start. How are you, everyone? How's things going on? How's the energy level today? Are you excited for this 2 days workshop, biochemistry? Awesome, awesome, great, great, great. So we will be going through a lot of basics to complex part of biochemistry today. Moreover, I want to take you to the cellular level of biochemistry also, and chemistry part of biology also. So both biochemistry, that is biology, is being deal with the chemistry part of it, that is your biochemistry. So we will be dealing with both part. Somebody was asking us about the certificates. Yes, you will get the certificates for sure by the end of your workshop. Um, by our Shivani ma'am and Vatsal sir, they will take care of it. So don't worry. And so meeting will be here only at 4:30 to 7:30. I will try to. Sorry for today. Just got 10 minutes late. But please join in. And it's not required to have your video on. So turn off your videos also. And some yeah. So it's almost 4:30 now. And one thing that is, I would like let people more people add that because they will miss it then. So just wait for 5 minutes more, then we start the session. I'm good. I'm good. I'm quite excited. I did this workshop once in last month, so I got a really good review from the students. So I want the same reviews from you also. I hope I could make up to your levels, to your expectations level. So what are your expectations from this workshop? Could you please write me down? What are your expectations? So I will share my screen. Yes, it's in the collaboration with them. So you will get the certificate. In We have a collaboration with them. Whereas myself, I will give you my little introduction just now. Who am I and where I am from, and yeah. So it is conducted with the cooperation with the IIT Bombay Excel ECEL. So, I hope everyone has joined us now. If so, a little bit about me. Who's your trainer? And so you can see my screen, right? Everything is being shared to you. Is Please just give me single thumbs up, 1 or 2%. Okay, that's good. So, and what about the voice? Voice is all good. Is it audible? Uh, I should decrease my fan's volume. Maybe that's creating some background noise. Just wait a second. Okay, so something uh, about me. So, um, I did my education as in my bachelor's BTech in biotechnology in 2009 from Shahid Udham Singh College in Chandigarh. Afterwards, I went for 5 months lecturership at Beant Singh College in Punjab, and I my thirst for education didn't stop there, and I want to have foreign experience. So I went to Germany for my masters in MSc in agrobiotechnology, where I switched from agricultural studies to animal studies, and I got chance to work with the Max Planck Institute of Heart and Lung Research, where I did my master thesis on the cancer metastasis. then after finishing in 2012 my master's, I switched to medical science, human medicine, where I did my PhD in molecular medicine, finished in 2017 under the University of Medical Center in Göttingen, where the highest Nobel Prize winners are known for, the city of Göttingen. And my topic for my PhD was redox regulation of PP1 and ER stress regulations of CTGF growth factor in cardiomyocytes. So, okay, I hope no one is want to get at more. So yeah, after having my PhD in molecular medicine, I came back to India, and then I joined as a assistant professor where I was teaching principle of biotechnology, genetics, and cytogenetics in the Department of Biotechnology. And side on side, I was also teaching. I was an online professor with Edufabrica as a freelancer for NEET also, and at the moment also I'm associate subject matter expert at Evelyn Learning. So everything, so that since that the days are changed for work from home, so things, your job perspective, your priorities also change then. And also I published one paper in Nature with scientific reports known as the reduced activity of PP1 alpha under redox stress condition. This paper is from my first part of my thesis. So this is something about me. So basically, I had a 7 years of staying experience in Germany, and then I came back to India. Now I have 2 to 3 years experience of teaching in the field of biotechnology and biochemistry. So, um, in order to do so, so before I start, what are your voice is not clear. So for others, is it clear, students? Okay, good. So please, bache, try to unplug your or turn on your computer on and off, and then try it again. Sometimes it's turn it on and off, make things work out. So, um, in order to go ahead, uh, I want to take you the journey of first of all cell. What is cell is, how small it is, and how big it could be. So I just want to start with the comparison of cell with you, from the rice grain to the virus cell, like how much size is the difference. So because we're gonna study about very minute things like amino acids, proteins, carbohydrates, right? RBCs, various biochemical diseases. So we should know first of all before we start some basics about cell. Because if cell is clear, then everything is clear for us. Okay. Um, so let me zoom. And beyond, before I start, also, it is not compulsory, but this is a workshop actually. This is not series. In series, we do that. But I would like to, before we start, I would be happy if you could send me on my email ID an assignment. Yeah, an assignment on my email ID about what we did today and what we do tomorrow. Just a two-page summary of everything what we do, whatever we learn, whatever we learn from it. You just make a two-page summary, and then you can yeah, send it to me, because this will be very helpful for you, because we will be going through many slides, many things on the link, but that will be beneficial only if you have learned out of something and write in your own language. You could write it in your own way, whether you want to send it by writing on a paper by your hands or by using wordpad, whatever you feel comfortable. If you feel like it's not compulsory to send me the assignment to complete your workshop, but I would highly recommend to do so. Everyone is doing this with me. So this is I did one workshop with biochemistry so far, and molecular biology and biotechnology techniques. Nowadays, I'm doing a workshop of plant biotechnologies. So everyone, students, they are sending me their everyday work, that what they have learned from us, in just like a two-page format. So in easy to understand, and this will be just for you, not for me actually. This will be for you, to keep it for the future perspective, that what you have learned, that you came to my workshop and you just didn't came for the certificate, because most of the students come to this workshop for the sake of certificates. But in my workshop, I want you to take some home message, that when you go, that Simran sir had taught you something, you have learned something, and it's beneficial for you. Any entrance exam, or any if you are going through your college exams, I don't know, you might have different needs of this workshop. So some home take message should be there always when you are leaving this workshop, with plenty of knowledge. So I think, if anybody has any questions actually, um, with this workshop or from me. If not, then I will start then. Okay, cool. So I will zoom in this very interactive explorer picture, where you can see the size of a coffee bean and grain rice, how much size is. This is a regular point, 12 point regular point looks like. This is a coffee bean by 12 by 8 millimeter, and the grain of rice is 8 by 2.5 millimeter. And let's zoom in and let's see where our cell stands for. So this is our sesame seed, which is 3 by 2 millimeter, then grain of salt, 0.5 millimeter, then amoeba now comes to the microbial level, that is 500 micrometer, then paramecium 210 by 60 micrometer. Now comes the human egg, which is 130 micrometer. Skin cell to be around 30 micrometer. The sperm cells to be 30 by 60 by 5 micrometer. The photoreceptors in the eyes, rod receptors, 2.5. Red blood cell, 8 micrometer. Baker yeast, 3 by 4 micrometer. Your chromosome to be 7 micrometer, like this. Mitochondria goes to 4 by 0.8 micrometer. E. coli 3 micrometer. So we're starting in the millimeter, and now see how things are going into micrometers, like lysosome just 1 micrometer. Now we are done with the micrometers. Now comes the nano. Nano is the viruses. So measles virus 220 nanometer, influenza virus 130 nanometer, HIV 130 also, rhinovirus to be 30 nanometers, hepatitis virus to be even smaller, 45. So this was something about viruses now. Now comes the next part, ribosomes, that is 30 nanometer. Further, uh, now comes the antibodies. They are all ribosomal RNA, transfer RNA, all the mRNAs, the RNA, different types of RNA. They are coming around to be 7 nanometer, hemoglobin to be 6.5 nanometer. Your lipids we will study today about, with the phospho and hydrophobic and hydrophilic ends, to be 0.9 to 3.4 nanometer. Glucose, now comes picometer. Now we are done with nano, now comes the picometer. So glucose to be 900 picometer, adenine to be 100 1300 to 760 picometer. methionine, an amino acid to be 1100 to 700 picometer, water molecule even smaller, 275 picometer, and then at the end your carbon atom, yeah, from which your everything is starting in this case is only 340 picometer. So you can see that how from picometer, so everything is existing in such a good manner and working so viably in a perfect zone that and happening in the milliseconds, and it's happening in the milliseconds. So someone has to join in. So from pico, nano, micro, millimeters. Just this interactive picture was to just show you how things are interconnected from the size perspective. They are within each of them, and but still they are working in a good manner. So that's it. So that was something about the cell, how within everything things are arranged, how much size is belong to what. Um, Now comes to the point is because I have many slides about your nucleotides, nucleosides. We will discuss basically today about DNA and its various parts of it. Because if we understood about DNA and its genes, we are able to understand its synthesis, its biosynthesis, and furthermore. So today is the day we will understand what biochemistry of DNA. In order to understand that, let me have this overview with you, students. So let me just start. Sorry for interruption. Let me just shut down this. So when the video will be playing, I might be not speaking. So, Bachche, the topics that we'll be covering today um, will be. So, today is the day one. So, basically, I will deal with the introduction to biochemistry, classification of amino acids, basic functioning of amino acids, lipids, nucleotides, and nucleosides biosynthesis and degradation. If we are still time is left, I would like to continue with the virtual labs. That will be helpful for your biochemistry experiments or even in the molecular biology experiments. So that things will be we will be covering for today. So this is a 10-day workshop, but I try to convert everything in 2 days because we take 10-day workshop for around 30 hours. But in our case, some topics will be dealt here from this topic, or some will be not. So I will try my best to have the most of the topics be covered. So something like lipids will be covered, amino acids will be covered. Um, and nucleic acids will be covered, yeah, enzymes will be covered. So these basic parts and virtual lab will be covered in these 2 days. So these two topics I will provide you one objective of like each day what we are doing, no worries. So before we start, just one interactive video to start things. So what are DNA and genes? Anyone, what are DNA and genes? Could you please let me know? What is DNA? Just type in the chat box. Functional units of the gene. hmm Is it genetic material? Okay. Anyone else? So, yeah, that is actually you got the right answer there. That is the genetic material that is passed on from parent to offspring to maintain genetic information and responsible for the variations. That's what I was looking for. It's a genetic material which passed from your parents to your further offsprings, which is the reason we have these variations happening in our body. So that's the DNA is. So, to something about it, let's start with this interactive video.
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order, but small differences in the sequence of the bases in our genes make each person unique.
So, that was it. That's the main reason that we have a difference of material in each part. So, let's start with the workshop with the presentations now. So, you have some overview now how cells are working, a little bit about DNA, but let's continue further. I will keep playing these videos in between if some terms are coming up. Oh my god, so sorry, somebody wants to get added, so I have to come back here. So please students, join in, don't come later. You already have half an hour left, so it's not good. um, so guys, get your copy and pen with you because this lecture is gonna be interactive. So don't worry that you will be getting these presentations that I'm gonna show you today. You will be having with you, but still try to make notes out of them so that you can make an easy. It will be easy for making you an assignment later on. Okay, I will give you uh, a lot of time now. Let's say 20 seconds. I will count to 20 seconds and get your copy pen with you, and then we start so that uh, we are all on the same panel. So I will start. One, two, three, four, five, six, seven, eight, nine, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. Okay, we are good to go now. Okay, Some glitch is there. Sorry for that. There's a glitch in the networking. Let me just solve it. Oh, oh. Okay. So welcome to the class. So we will be dealing with the some basic biomolecules. What is that biochemistry is all about? Let's continue. So from this chapter, you will be understanding how behavior and structure of complex molecules found in the biological matter. So the way these molecules interact to form cells, tissue, and whole organisms. So we will be dealing with all these parts in this case today. So, what is biochemistry? Anyone? What is biochemistry? So, could you please write me some definition about biochemistry? Chemical change in biological system. Okay. Mm-hmm. Assimilations of both chemical science, biological science. Oh wow, even advanced. Subject involves biology and chemistry. Mm-hmm. Mm-hmm. This is a very quite near to the statement that's I was looking for. So that's we are studying today. Branch of biological science that deal with the molecules involved in the biological even more apt. That's good one. That's really good one. So biochemistry is the application of chemistry to the study of biological process at the cellular and molecular level. So we are bringing the chemistry part into this to the biological process to study everything at the cellular and at the molecular level. Okay. And it has emerged many different other disciplines around the beginning of 20th century when the scientists combined chemistry, physiology and biology to investigate the chemistry of living systems. So they combined chemistry, biology and physiology, all three different disciplines to investigate further like in 20th century. So what is the chemistry of living system is looks like. So principles of biochemistry. So basically, cells, that is a basic structure unit of living organisms, are highly organized and constant source of energy, and is required to maintain the ordered state. Yeah. So living process contains thousands of chemical pathways. Precise regulations and integration of these pathways are required to maintain life. So certain important pathways such as glycolysis is found in almost all organisms, and these organisms use the same type of molecules such as carbohydrates, proteins, lipids and nucleic acids. And further, with these molecules they give instructions for further growth, reproduction and development of each organisms, which is encoded in their DNA. Guys, please don't come so late. You are getting more than 30 minutes late. So this is very horrible situation then. So I have to stop in and keep you at. It's like in the class. So may I come in, sir? And we are like, okay, you're 30 minutes late and lecture is almost over. So what shall we do with you? So we let you in. Actually, at the end, we want that that whatever the information that we are presenting, you can also have in the last minutes. Only if you're there to perceive it. Otherwise it's have no use. So see, there is one more person like that came again. So please add on time. It's difficult for us. So okay, so that was basics about biochemistry. What is the basic definition and basic principle behind it? That is, it's deal with the your chemical pathways to integrate and maintain the life with various cellular pathways using various molecules, so that it could maintain the growth, reproduction, and development of the organisms in order to maintain their DNA. So cells, we have just recently saw that how basic cell size goes from smaller to biggest, and the basic building block, they're the basic building block of life actually. They are the smallest living unit of an organism, which could grow, reproduce, use energy, adapt, respond to the environment. Many cannot see, we cannot see them with the naked eye, so we need a microscope to observe them. Okay, so I will not add that person anymore. A cell may be an entire organisms, or it may be one of the billions of cells that make up the organisms. Basic types of cells uh, are two types here: eukaryotic and prokaryotic. So you can see left and right, how the different morphology is of these two different cells, and how the different inside the holding genetic material is also different in both cases. So here you can see in the eukaryotes, let's start from the Golgi body having the vesicles, then there's a plasma membrane, and then there's a nuclear envelope outside of the nucleus, then outside the nucleus there is endoplasmic reticulum, inside is the nucleus, then is the cytosol, cytoplasmic things present, and cytoskeleton is present with the lysosome and also peroxisome and mitochondria. On the other case, in this side we have a bacteria with the flagella, pili, an outer membrane. We have cell wall, proper periplasmic space, plasma membrane, cytosol, ribosomes, and nucleotrigens. So these are all. That's how the basic difference is between both. My screen is not visible. So is it visible for others? So maybe try to. Yes, it is visible to others. Dibya, try to switch it off and try to get in again by using the link again. So maybe you can see the things back. Or click on my screen, something you might be not clicking on our screen. So that's why there's a difference. Okay, all good. So cells. So we have discussed this. So nucleate regions contains the DNA, that is cell membrane and cell wall, and contains ribosomes to make proteins in this cytoplasm. That contains three basic structures: nucleus, cell membrane, and cytoplasm with the organelles. So that is your prokaryotic structure. On the right side is the eukaryotic structure. So we understood the main difference between them that we have did. Then comes the essential part of biochemistry, that is your water. So it's about 60 to 90% uh, percent of your organism is water, where water is used in most reactions in the body, and water is called a universal solvent. So now comes the major biomolecules that are used in the biochemistry subjects here in the field of biochemistry. So just like cells are building blocks of tissues, the same way molecules are the building blocks of the cells. Same way molecules are the building block of the cells. So there are around 10,000 kinds of molecules that are present in the animal and plant cells itself, and these we call them as biomolecules. So water constitutes around 50 to 95% of cells containing content by weight, and ions like sodium, potassium, calcium may account for another 1%. Whereas the major biomolecules are your CHNOPS, that is carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. An infinite variety of biomolecules contains carbon mostly, and most biomolecules considered to be derived from hydrocarbons. Mostly we get it from the hydrocarbons, and the chemical properties of organic biomolecules are determined by the functional groups. That is, most biomolecules have more than one functional group then. So that's how you can differentiate between the different chemical properties of various biomolecules. So building blocks, they are the building blocks: simple sugar, amino acids, so nucleotide and fatty acids. So these are the various respective their biomolecules, that is macromolecules, polysaccharides, proteins, RNA, and lipid respectively. So fatty acids are the lipids, nucleotides are the RNA or DNA, amino acids are the proteins, simple sugars are polysaccharides, building blocks are your macromolecules. So basically, we are just touching the basic, very basic part of biochemistry, and this one from here, going from your macromolecule to the building blocks to the left side, it is a catabolic process that becoming from smaller to bigger part. But if it is going from bigger to smaller part, that is your anabolic process. So that's how there is a difference between catabolic and anabolic process. Then there are linking monomers. So you can see a sugar here with the six carbon, this benzene ring, kind of a benzene ring, but not, it's not benzene, but six carbon and a hexagon. So where you have hydroxyl group attached to it. So what you do to monomers, you want to join it. So you need to dehydrate. Removal of water has to be there. So H2O need to be removed to join them together. So this process of joining two sugar monomer to make a double sugar. So we remove H from this and we remove OH from this. Okay, and then H2O form out of it, and then breaking down of polymers. So cell breakdown polymers process called hydrolysis by adding a molecular water. If we add a water molecule to break the macromolecule, that process will be known as hydrolysis. So like this, we have two different macromolecules joined together, but we added H2O, and then by this there's a breakdown of the polymer with the OH and OH like that way. So in the earlier case, we studied about joining of it with the help of H2O by dehydration synthesis. Whereas in this process we understood about hydrolysis. By addition of water, we break the bonds between two macromolecules. So water added to the split a double sugar. So double sugar molecule has been splitted into two different parts. Further, now comes in the macromolecules is sugars. So most abundant organic molecule found in nature are the sugar ones, and they are initially synthesized in plants from complex series of reactions, basically involved in the photosynthesis. And basic unit is monosaccharides, and so basic functions of your sugar are to maintain energy, to store energy in the form of starch or glycogen. In the case of animals, it's glycogen, and in the case of plants, it is starch that is used for photosynthesis in plants. Further, they provide energy through the metabolism pathway and the cycles, and supply carbon for synthesis of other compounds, and form structural components in the cells and tissues, and intracellular communications also. So you might see that these sugars are really important component for students also. You have sometimes, right, sugar craving when you have a lot of studies going on that your brain needs some glucose externally. Why it needs? Because there is a lot of uh, sugar getting decreased with the increase in of your brain usage in your body. So once you had a bar of chocolate or anything sweet, you feel very energetic, your brain feels really good afterwards. So that is where biochemistry is playing role. But don't eat excessive chocolate. That is something called like giving boost for the temporary reason. Instead of having a sugar, try to go for a green tea or some other healthy or alternative, because too much sugar is also not good, but sometimes it's good, it's healthy. If you're really craving for chocolates, also try to go for dark chocolates, which are now available in India very easily, and rates are also similar. Instead of going your normal Cadbury and 5 star, they are not chocolates at all actually. It's just sugar, lot of sugar. So go for dark chocolate where it will be bit bitter, but lot of your good hormones will stimulate for your studies if you have to study for the long time. From the study, I just thought that could you please introduce yourself, from uh, which part of India you all are actually. So what are you studying, so that I could be a bit aware of my students. So I'm teaching to this part of India, because that's the beauty of this COVID actually. That's a I should not say the beauty, but we India is such a diverse country. We are still quite united, having a diverse background, and we are on the main focus during these workshops, just to have good knowledge, good time, and to have this knowledge for us with most of the time. Yes, Bache, Deep Narayan, we will give you the presentations just like that, so that you can keep it for yourself. Oh, from Nagaland, welcome from East India also. Uh, I want to visit Assam once, Sikkim also, but it's just okay. West Bengal, Rajasthan, okay. BTech Biotechnology, Xavier's, okay. West Bengal, Uttarakhand, wow. Nainital, wow. Banaras Hindu University, wow. It's such a beauty, wow. Just interact within the group with each other. Maybe someone of you could help out each other in their career progression. Just be friends with each other from after this workshop, and I will be in touch with you also if you need any questions. If you have any query regarding moving Germany or having studies outside, you can ask me anytime. Feel free to do so. Uh, or any other career questions. Shivaji University, Kolhapur, Maharashtra, Bareilly, Uttar Pradesh, Odisha, Delhi. I'm from Delhi, guys. Hi, Delhiites. These presentations are going from Delhi. If something goes wrong, Jorhat, Assam. Okay, I gave one interviews in Jorhat. You have recently opened one COVID institute there in the CSIR institute. So I was there for online interview. Um, but it was actually some other results came out not positive for me. But I would love to visit Jorhat, Assam. Calcutta, great. I have a lot of tenants from Calcutta here, a lot of um, friends from Calcutta. You, okay, good. DU, Delhi University, nice. Okay, so you can see in the chat itself, you're from very diverse part of India, and still we are working on the same working for the same aim, that is to learn about from these workshops. In order to do so, let me start with another video here. So same gene and different functions, or different organisms. Sorry. So so two things will be having same gene and showing different organisms. How is this possible? How is this even possible? So to understand that, let's go through this video then.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing. Animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
So beautiful story how GFP, your green fluorescent protein, was present. Uh, Let me just stop this. How it's present in the jellyfish, and we can make it also present in our other organisms to see their effects, and also the spider web, the silk web that we were using to construct a really good hard silk actually. So from different organisms and presenting using it for the different stuff. So we were using for the biofermenters and producing in the large scale. So these all being all possible, even though they're the same gene, but in different organisms, they are doing the different functions. Yeah. So that was something about same gene, different organisms. So shall I share all these links while we are going on Google Meet or on the WhatsApp chat? So that you can keep it. Maybe the WhatsApp chat will be better because after this Google Meet is over, things we will be not able to keep it with us. So I keep sharing things what we are going through uh, here. So I keep sharing so that we are not in lost afterwards. So please, when you are done with the lecture of today, go through these pointers. These are various pointers just to have a flow of the workshop. That first we understood about the cell, then we understood about DNA, genes, then we understand same gene, different organisms. So everything is interconnected with each other. It's like I'm trying to solve a puzzle because it's so vast, and I don't want to make it quite boring for you. I want to make it interactive. That we also do learning from the presentations, and we also go through some videos and lectures, some interactive sessions, so that all these small things at the end of our workshop, they make a sense. They make a sense that why we are here for and what we have learned out of it, making a what we say impression for yourself, or also about biochemistry, what is biochemistry was all about then. So to continue, um, yeah, we are done with the sugar. Now in the various we were talking about the biomolecules. Now comes the other part, that is your fatty acids. uh, Fatty acids includes mainly your lipids. They are the sorry they are your monocarbolic carboxylic acids, contains even number of carbon atoms. There are two types, that is saturated and unsaturated. In saturated there is a single bond, in unsaturated there is a double bond. And fatty acids are the component of several lipid molecules. And example are lipids of triacylglycerol, steroids, fat soluble vitamins. All these includes your fatty acid lipids. And whereas your polar hat hydrophilic is this one is your hydrophilic end, this side, and here is the non-polar tail, that is your hydrophobic end. So this one is water loving. Philic means loving. Phobic means to get afraid of. So functions are to storage of energy in the form of fat, membrane structures, and insulation of thermal blanket. Further is synthesis of hormones is also done with the help of fatty acids. Then comes your steroids. So these two steroids shown to you that they are the carbon skeleton of steroids is bent to form four fused rings. So mainly in the steroids, they are the four fused rings always present. Immer, immer, immer. Always they will be present in them like this. Yeah, and there you can see the main difference is on the side chain of here with the hydroxyl group. Here is a double bond O, and there is a hydroxyl group OH. Then methyl group is constant in both cases, in three cases. Here is also methyl group is constant, but here we have a long chain here of alkyl acid, and here's only hydroxyl, and there is a double bond. So somehow these got interchangeable in this case, and we have a big bond there. So this one is your cholesterol, and cholesterol is the base of steroids from which your body produces other steroids from cholesterol. That's why this triglyceride triglyceride acids TGC. So you go for your test in the morning. They say you have to check your cholesterol. If your cholesterol is not balanced, so maybe your other steroids in your body they are not balanced. So you have to maintain that also while doing so. And estrogen and testosterone are also steroids. So the left one was the testosterone and right one was the estrogen in front of you. So you understand that and most of the gym people they are taking testosterone steroid hormone externally to build their muscles, which is on short run is okay if you're somebody of you is going for competition in the bodybuilding, but on the long run taking steroids has very drastic effects on you because your body is meant to run on the normal level. But if you want to increase your body muscle mass to increase the muscle of a body, then you need some extra steroids than the normal body has. So in order to do so, you need to maintain it by taking some extra steroids from the outside to do something extra for your body, because your body will not produce anything extra without taking anything extra. So that's why all the bodybuilders they are taking testosterone hormones to increase the body thing externally. Also, I mean, from the exercise point of view, one thing more I got to know, who are of you running, you might see that the first week of running will be really hard for you. It will be very worse condition for you if you are going for running, because your body in your muscles of your legs, you will have less ATP production, less mitochondria production. You will have very less. What do you say? Um, yeah, the less energy is being produced. There's a lot of stiffness in your muscles, and this is why this is happening. This is happening because of less energy being produced in your cells, because earlier it was used to work in normal walk. So it was just walking within the house or going for some work, or driving on a scooter, or sometimes maybe cycle, not much. But once there's a less part of mitochondria which is producing less ATP, so suddenly after 1 week it will understood that you need to require to produce more mitochondria, and more mitochondria means more ATP. So you will build new muscles for that. So your stomach, your legs muscles will be produced in a more distinct way. It will be produced, muscles will be produced for your running sessions. So you will, if you are running 1 hour each day, you will have muscles for your legs running for one each day, and to give you energy to run for 1 hour, because nothing will work for you as in just in a day for you. So you will have a pain for few days, even for any exercise that you're doing. You will have a pain for few days, but later on they will be fine with you, because extra mitochondria and ATPs are producing in that case. Nucleic acids. So nucleic acids are one which store hereditary informations, contain information for making all body proteins. There are two types of nucleic acids, that is DNA and RNA. Then amino acids. Amino acids are the one with which we known as a protein. So it has one alpha hydrogen, one alpha carbon in center. On the side is the alpha amino group is attached with R group side chain on the top and carboxylic group COOH being attached on the side. So this is how your amino acids basic carbon atom, sorry, amino acids basic molecules looks like, and it's a base building block of proteins, your amino acids. The R group determines the chemical properties of each amino acids, also determine how the protein folds and its biological functions, and functions as a transport protein, structural proteins, enzymes, antibodies, cell receptors. So proteins as enzymes. So many proteins act as a biological catalyst or enzymes. Thousands of different enzymes exist in the body. So enzymes which control the chemical reactions by weakening the body bonds and lowering the amount of activation energy needs for the reaction. That is, they are known as a catalyst. No, not to interfere with the equilibrium of the reactions. Enzymes are reusable. Uh, so to understand about enzyme now, they have an active site. So which is a cleft or groove in the enzyme that binds a substrate of a reaction. So there is always a cleft or groove in it, from where you have a bind or substrate of reaction. Nature and arrangement of amino acids in the active site, and it's specific for only one type of substrate, and they accept just one enantiomer. So here there's on the active side nothing came, substrate didn't came, but on this side this is a egg white lysosome. Here your substrate came and make your protein active by joining the substrate of your reaction. So what did you understand in this lecture here is that you understand the basics of biochemistry, various biomolecules of the life, and types and functions of these various biomolecules. So these are the references. So you will be also provided with the PDF books of biochemistry. I think it's not possible to send you books here in the big with the big files as of now. So how can I send you books if I want to send you books and these presentations? What do you recommend? On which platform shall I send you? Telegram app. So yeah, I can see if we can try the Telegram. We'll add you all onto the Telegram and can send you some books also PDF. I have Lahinger also, Lippincott and Harper. I have three major books. So I try to do so on that. So guys, I will take a 5 minutes break. I see you in 5 minutes then, and we start with the next lecture. Okay? Or if somebody could make a group, I could share the group, then maybe something like that. We will figure it out later, don't worry, we will make it happen. So guys, uh, okay, drive link and share it with you. Okay, this is also good way to do so. The Google Drive. Yes, yes, I will try with that also. Okay, guys, let me come in 5 minutes. Just have a break, have a Kit Kat break, have some water, then see you in 5 minutes. Take care in the meantime. See you soon. So guys, I'm back. Try to do live streaming also for you so that I could record it. But I think it's, it stopped. Anyhow. So guys, you will have for sure the presentation, but recorded version is not possible. I tried to do live streaming with YouTube also today, so that I got some messages. Somebody's couldn't attend the workshop today, but for the later maybe you can check it. But let's see if it works out or not. Uh, so I will check the Google Drive for you. If I could make it Google Drive function for you all people. So okay, there was some sounds coming up. I just want to check. So let's continue uh, in another lecture. My computer got really slow now for some strange reasons, which is not good. So we have talked about the basic biomolecules in the case of uh, biochemistry. Now we're gonna discuss about various amino acids and its properties. So let's start. So in this objective, you will understand about introduction to the amino acids, optical properties, further acid-base properties and buffers characteristics, physical properties, importance and functional role. So all these five things we will be dealing in this amino acids. Um, so before we start about okay, we continue new with this presentation. Then I will take you to the interactive section with the protein side also. So, what are amino acids? They are the building blocks of proteins, which has general structure as we have discussed previously. You have alpha carbon between which carboxyl and amino group are attached. So, your amino group and carboxyl group are attached, common to all the alpha amino acids of the carbon. You also have hydrogen attached to it, and on the other side is a side chain which makes it a different functions. You have different kinds of amino acids just because of this different distinctive for each amino acid, this R1. So building blocks of proteins, they are the building blocks of proteins. General structure of amino acids includes alpha amino group, which has alpha carboxylate group and variable side chain, that is R in this case. So amino acids as a acid and base, as the acid amino acids contains ionizable groups, so that they cannot act as an acid and base in a solution depending on the pH of the solution. Due to this nature of amino acids, they are called as deuterions or dipolar ions. And they do not have any charge but behave as acid or bases depending upon the pH changes in the solution. So they do not have charge, but they could be acidic or basic depending upon the pH, as how much is the pH is. And they are, because of that, they are act as neutral ions, or we can also call them dipolar ions, because they can change their nature of their basicity or acidity. Just how much pH is of your solution, they change according to that. So biological importance of amino acids: over 200 amino acids occur in nature, but only 1/10 of these occurs in proteins. The biological functions performed by them are building blocks of peptides and proteins. As precursor, many biologically important molecules are derivatives of amino acids. For example, tyrosine is the precursor of the hormone thyroxine and skin pigment melanin, whereas the tyrosine is also the precursor of a compound abbreviated as a DOPA, that is dihydroxyphenylalanine. So there could be also precursor in that case. They could be also building blocks of the peptides, and yes, hence they could do various functions. On the other hand, furthermore, to add on this, they could be act as a neurotransmitter, that is transmission of your impulses in the nervous system. For example, tryptophan is a precursor of vitamin named nicotinic acid, your vitamin B3. So tryptophan could also be uh, precursors of many vitamins. They could be source of sulfur derived from sulfur containing amino acids. They could be involved in many metabolic pathways such as gluconeogenesis, where it is involved in the glucose synthesis. In the glucose synthesis also, amino acids plays a big role. So what are the physical properties of amino acids? So amino acids are mainly water soluble because of its polarity and presence of charge groups. But they are also soluble in the polar solvents, but not soluble in the non-polar solvents. The one which are non-polar, they will be not soluble in that case. They have very high melting point, reflecting the high energy needed to break the ionic forces, maintaining the crystal lattice. On the other hand, they are colorless crystalline substance soluble in water, and their melting point is above 200 degrees Celsius. So their optical activities, it is exhibited by all amino acids except for the glycine, as it does not have any asymmetric carbon atoms, whereas the uh, dextra rotary positive isomer, which has the ability to rotate the plane of polarized light to the right, and there's a laboratory negative, which has ability to rotate the plane of polarized light to the left. So both isomers can rotate the plane of polarized light by the same magnitude, but in the uh, opposite direction. Other properties of amino acids are, so amino acids, base properties of amino acids is that it has two ionizable groups, that is your alpha amino group and alpha carboxyl group. So some contains additional acidic or basic groups in the side chains, which are responsible for acidic and base behavior. So as a result, their ionizability of the following equilibrium can be written as, so if you have this carboxylic group added, COOH, so H could be removed, or from NH2 H could be added. So in this way, they are become zwitterions of your alpha channel. They could be negative and they could be positive by removing the hydrogen and addition of hydrogen. So since these are reversible, these reactions are reversible, and they can act as acid as well as base, which explains them as an amphoteric property of amino acids. That is, having an acidic and basic characteristics, they are known as amphoteric property of amino acids. So what are zwitterions forms? What is a zwitterion ion forms? As we discussed, all neutral amino acids are present in the zwitterianous forms at physiological pH around 7.4. The carboxylic group will be unprotonated and the amino group will be protonated. So this is how your zwitterionize forms are formed. So amino acids can be present in two configurations, the L isomer and D isomer, which are mirror images which differ in the arrangement of the chemical groups around the alpha carbon atom. Okay. So you have L isomer and D isomer, which are mirror images of each other, like this. In this case, you have CH3, COOH, COH, alanine group, as we can see. So amino acid could be present L or D isomer, and all amino acids present in the L configuration as L amino acids, and D would be your D amino acids. Most of the time, they are present in the L configuration only, like this one. And this is how your enantiomers are being made. Further, the chemical properties of amino acids, reactions due to the amino group. So this reaction is called as ninhydrin reaction. Ninhydrin is a powerful oxidizing agent that decarbo decarboxylate the amino acids, yielding carbon dioxides, ammonia, and aldehyde. So it removes from the amino acids, it decarboxylate, and it removes carbon dioxide, CO2, your ammonia, NH3, and aldehyde group. So the reduced ninhydrin reacts with the liberated ammonia. So ninhydrin, the reduced version, will react with the ammonia to form a purple color complex called Riemann purple. So it forms a Riemann purple, and color direction, and further chemical properties of amino acids. We can see that amino acids can be identified by specific color reactions like xanthoprotic test. So in this, by benzene ring of aromatic amino acids, we can check with the help of Millen reaction for the presence of phenolic group of amino acid, that is tyrosine groups, or sulfur test for sulfahydryl group amino acids, an anhydrin test for alpha amino acids. So these are the various tests you can do. Millen reaction for the presence of phenolic group, sulfur test for the sulfahydryl group, anhydrin test for the alpha amino acids. So you can check all these tests, for xanthoprotic test for the benzene ring of aromatic amino acids. So these are the various color direction tests. So that was about to check your chemical properties of amino acids. Further, the chemical properties of amino acids includes reactions due to carboxylic group. Amino acid forms salts with bases of esters with alcohol. Uh, and decarboxylation is happening, undergo the decarboxylation to produce corresponding amines. They are producing amines because of decarboxylation, and the reaction is quite important in the living cells due to the formation of many biological important amines like histamines, tyramine from the amino acid, and histidine and tyrosine. So what you have understood from these lectures is that you are able to understand the basic building block of proteins, that is amino acids, how they are the biomolecules of the life, and further you are able to understand the various physical and chemical properties of amino acids, which are help in understanding their metabolism, which help into understand their metabolism. So that's it. So we are done with the amino acids and its properties. So students, there is one more workshop that we will be running, that is molecular biology and biochemistry techniques, that we have recently been successful for last 10 days. So please try to contact Shivani ma'am. The registration is 1000 rupees if you're joining us for the project. If you want to do internship, then respective registration is there. So please contact with them if you're interested in joining the various techniques by staying at home, having a very strong like a certificate that you want to get which could lead you to the job in the future. Of that I'm very sure, because some of the students also published reviews in their journal on their own. So just look for that. So any questions, students, any doubts? All good so far? If yes, let me just, because there's so many things are open, so my computer is hanged. Sorry guys, just a little bit delay. So let me close all the things before. So let's build a DNA. We, when we come to the part, so go for the selection. Subtract connections, which it's at. So let's continue then. If there are 6 cysteine residues in a protein, then how many sulfur bonds are possible? So it's depend upon your protein conformation, because if the two cysteines, how can I split it? So somebody asked a question that there's 6 cysteines present in a protein. So how many they make these cysteines double bond with each other? That was actually my thesis topic of my PhD, disulfide bridges. Um, Let me just attach my pen to this, Viacom pen. So it depends upon your protein actually, how is your protein conformation is. So let's say if your that is the conformation of a protein, and this is your cysteine is present. This is your cysteine. Then one cysteine here. You said 6 cysteines, or one cysteine here, one cysteine here, and one cysteine here. So they could make disulfide bridges like this, like this. So so there could be 4 bonds. But if the cysteine is not present here, they are present somewhere else in these. So they are present here, but the vicinity of these proteins are quite far from each other. It's not possible to make disulfide bridges. So that's depend upon where your protein is lying, and then it could make the disulfide bridges there. That's it. I hope I could have answered your question, um, Avnita and Rita. Sorry. Okay. So in this topic, we will be covering about disciplines that we learn from various classes of amino acids. Explanation of different types of structures and their role with respect to amino acids. So they are the molecules containing amine groups that we have discussed. Carboxylic group side chain. The key elements are carbon, hydrogen, oxygen, nitrogen. They're the structure building units of proteins and other molecules. They're also utilized energy source. So uh, there are two types of essential and non-essential amino acids. Essential and your non-essential. So there are 20 different amino acids in total that make up the proteins of the human body. The amino acids that are needed to maintain replenished tissue, red blood cells, enzymes, and other substances. So 9 to 12 can be manufactured by the body. Non-essential amino acids not obtained from the diet. So body could produce these non-essential. The remaining 10 to 11 essential amino acids must be obtained from the diet. The rest of them we need from the diet, from the essential diet, and the rest we need from the body. And also some people for their exercise they are using uh, taking whey protein, extra whey protein outside from the body to increase the body mass, which is also good to some extent. But to my knowledge, if you want to maintain a natural body, if you are into the study thing, then you should take care of taking external enzymes. Like people also taking glutamate, alanine, many other enzymes in their diet in order to build their mass of a body, which is good for to some extent but not on the long run actually. So take care of that. So these are your following non-essential amino acids. They're all non-essential amino acids: alanine, uh, asparagine, aspartic acid, arginine, cysteine, glutamine, glycine, glutamic acid, serine, proline, tyrosine. So all these includes in them. Then essentials include your isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine. So you can remember them also by like I, LL, M, PTTV, or try to remember them like making sure these long terms into some meaningful name, some sentence, so that you can remember these forever. So you can create like I did LLM on Pakistan television Chile TV, or Pete Pakistan Tele Network TV. So I had LLM Pakistan TV something you can into it. A triple four triple A of times A 4A, then C, then triple G, and CPT. This is how you can remember their name. 4A, 3 C3G, SPT. So by this you can remember all the name of your non-essential amino acids. So it's a quite easy way to remember. This is how you can remember many things. Sorry, many things for your reactions. Okay, the functional classification now, that was between essential and non-essential amino acids. Now comes the functional classifications. It includes your anabolic and catabolic responses, tissues, and pH regulations, glutamic acids, glutamine, which regulates your catabolic and anabolic reactions. The urea cycle and nitrogen management is done by the arginine, aspartic acids, and asparagine. So these are functional classification of amino acids. So essential amino acids for the protein and energies are isoleucine, leucine, valine, threonine, histidine, and sulfur-containing amino acids for methylation and glutathione reactions are methionine and cysteines that we use mostly. Neurotransmitters and precursors, so phenylalanine, tyrosine, and tryptophan. We use all these for their reactions, and they are precursors to heme nucleotide cells, and they are glycine, serine, and alanine. And bone collagen specific amino acids are proline. So somebody's speakers got on by mistake. Please turn it off. Somebody's microphone, sorry. Just a second. Okay. So we are done with essential, non-essential, functional classification so far. And now with the chemical structure of amino acids that we were talking for so long. So here you can see is Glycine is there, right? Which is just your normal with H and H on top. There is no methyl group. But when methyl group is attached, it's a alanine. So we're talking first about non-polar compounds. Then is your valine. Valine has three methyl groups. Then comes the cysteines. Cysteine has a sulfur group at the bottom. And remember, methionine has one sulfur group also here between your two after two methyl groups. Like this is a ethyl group, then there is a sulfur group, then there is a methyl group. This is your methionine. Then proline is your attached ring. It's kind of a ring. It makes this proline. Then comes the leucine here, CH2, CH2, CH3, CH3. Then this is your isoleucine, making a different way of structure of it from the leucine. Then tryptophan has these rings in it. Then phenylalanine, look at this. Okay. So there's a polar ones. Polar ones are there. The OH with attached to it, then it's a serine. If it is OHCH3, then threonine. And if a tyrosine is attached like this, asparagine to be here, glutamine to be here. Look like this. So there is a negative and positive charge. Then positive charge are the lysine, arginine, and histidine. So all are having amino group attached to them, which is giving them the positive charge. Whereas the negative charge are with the carboxylic group, aspartic acid and glutamic acid. So at the end of their methyl group, there is a carboxylic group is attached, and this is they are known as to be negatively charged group. So they have quite a good number of uses in human nutrition. When taken up in the human body from the diet, the 22 standard amino acids either are used to synthesize proteins and other biomolecules or are oxidized to urea and carbon dioxide as a source of energy. The oxidation pathway starts with the removal of the amino group by transaminase. The amino group is then fed into the urea cycle, and the other product of the transmutation is a ketonic acid and enters the citric acid cycle. Whereas the glucogenic amino acids can also be converted into the glucose through gluconeogenesis. So non-protein functions are they are quite many amino acids are used to synthesize other than molecules. For example, such as tryptophan is a precursor of the neurotransmitter serotonin, whereas tyrosine is a precursor of the neurotransmitter dopamine, and also glycine is a precursor for the porphyrins such as hemes. Arginine is a precursor for the nitric oxide, and aspartate, glycine, and glutamine are precursor for the nucleotides. So these are the various non-protein functions. So what is the outcome? So we are acquainted with the basic classification of amino acids and understand their basic structure. So that's it. So I hope you have understood. So how can I switch this off? Okay. Everything is slow now for strange reasons. So yeah, I will send these slides. Don't worry. No, this is without internship, so this will be not an internship included. So in 2 days it's not possible, but 10 days one will have. So there is some surprise to you. So which will be very. So just ask Shivani ma'am, ask the persons to be in touch with you. So shall we go to the next part? Uh, that is lipids. Before we go lipids, let's have some interactive sessions by a bit. Yeah. So you all know how DNA is made. So what are the basic, what is nucleotides? Anyone tell me what is nucleotides and what is nucleosides? What is the basic difference between nucleotides and nucleosides? Yes, sides are with the sugars and bases. In the tides, there's a phosphate group. That's the main difference. Great, great. So we are good on this part also. With this part, by creating a DNA molecule. So with the T, what comes in front? GC. In the T amino group, adenine, yes. What should be the base pair to the T? It's A, yes, like this. The T, it would be A. Then for the G, it would be T A. So by this way, your whole DNA will be made. T would be A. Then for the C, it would be G. For the G, it would be C. And for the A, it would be T. Like this, it will keep going on. So your DNA will keep going on like this. G, G, A is T, G is, oops, sorry. I, I just, my thoughts went to somewhere else. A is G, G is C. So till when we have to do this? A. So at this rate, we will need, as I've asked, when till when I have to do this. So it's told me that I have to work for 50 years, day and night, to transcribe the DNA in one human cell. So for the next 50 years, I keep selecting A and T and CG. So my whole DNA could be transcribed easily in that manner. So as simple as that. So try this thing in your free time. So maybe it took you maybe less than 50 years, not 50 years. Go for it. So this was very nice interactive session. This was Bache, these are interactive explorer which has been done by the Utah company, Utah University. So they have a very nice website, Learn Genetics. So on this, they put everything to understand these things on from the basic level, these assignments, so that we can understand what is the molecular biology, what is the main definitions for you. So I'm just check your just check your mobile. I'm sharing it on your WhatsApp group, all these animations. Check them so that you can use them, watch them later for your also, for your benefits also. So I will switch this off. Now, as we have done with the amino acids and protein, let's have one interactive session about proteins, what are proteins are actually. So let's have one video for that. So I will
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
So I hope it's clear that what we did so far essential to make proteins, and from proteins our body function is happening at a good rate, at a good pace. So that was the main reason about what our proteins is. So I share this also. Mhm. So next, transcription and translation. No, not now, maybe later. We can go through this video later. So first, uh, go through the lipids. So um, yeah, so we in this part I'm going to deal with the various biochemistry of lipids. So we are done with the biochemistry of amino acids and other stuff, other biomolecules. Now comes the basic part of your lipids. So they are neutral lipids. Uh, that are lipids which contain hydrophilic and hydrophobic groups, that is your water attracting and water exposing, that is the water attracting are exposed to outside, and the heating group that are immersed interior. So that's why they are neutral lipids, and they basically are triglycerides made of three fatty acids linked to a glycerol and have no polar group as a phospholipids do. So although they may have hydrophobic tail, they do not have hydrophilic head. So phospholipids are the complex or compounds lipids containing phosphoric acids in addition to fatty acids, nitrogenous base and alcohol. So there are two classes of phospholipids, that is glycerophospholipids, glycerol plus alcohol. Further is sphingophospholipid, that is sphingosine and plus alcohol. So this is how the two classes are of phospholipids sounds like. So, glycerophospholipids, they are the major lipids that occur in the biological membrane, uh, and they consist of glycerol 3-phosphate esterified as C1 and C2 with fatty acids. And we have one of them, the glycerophospholipids example is phosphatidic acid. So this is the structure of phosphatidic acid, where in one carbon you have carboxylic group with a alkyl group attached to it. Then other to the two methyl group, again one carboxylic group, one methyl group is attached to it, R1. Then there is a phosphate group attached to it. So this is how your phosphatidic acid groups looks like. And they are simplest phospholipid. It does not occur in good concentration in the tissues, and phosphatic acid is an intermediate in the synthesis of triglycerols and phospholipids. And other glycerophospholipids containing different nitrogenous bases or other groups may be regarded as the derivatives of the phosphatidic acid. So there is also lecithins in the lipids. They are made up of phosphate plus choline. They found in the egg yolk. Lecithin is mainly found in the egg yolk. So it is a phosphatic acid with choline as the base. They have choline as base attached to it. And like this, we have the CH2, CH2. Then this one. On the other side, we have this choline attached to it on the phosphate group. Like in the previous, we have this only phosphatic acid, and phosphatic acid choline group has been attached like that on that case. So phosphatidylcholines represent the stage form of a body choline. They represent the body of the choline, and it shows like this. And cephalines, ethanoamylene is the nitrogenous base present in the cephalines, and phosphoric acids, ethylene, amylene are attached to it. So further, phosphatidylene, serine, phosphoric acid plus serine, amino acid serine is present in this group of glycerophospholipids, and phosphatidylene theorine is also found in certain tests of this. So plasmogens, when a fatty acid is attached by an ether linkage group at C1 glycerol in the glycerol phospholipids, they resulted in the plasmalogens, whereas the cardiolipins are the ones that are isolated from the heart muscles. And structurally, a cardiolipin consists of two molecules of phosphatic acids by an additional glycerol through the phosphate group. So functions of phospholipids, they are mainly structurally component of membrane, regulate membrane permeability. It's participate in the absorption of the fat from the intestine. Furthermore, they are essential for the synthesis of different lipoproteins, participate in the transport of lipids. So cholesterols are found in animals and it's called as animal sterol, and 1 gram of cholesterol consumes per day in our body, and functions are such as provide integrity to the cell membrane, biosynthesis of the bile acids. So biosynthesis of cholesterol is how is it happen? They are the source of energy ATP, and the precursor is the acetate of acetyl coenzyme A. Their biosynthesis is basically done in 5 steps, 5 stages. First, there is a synthesis of malonate from acetate, 6 carbon chain. Then there is a synthesis of HMG coenzyme A. Then production of isoprenoid units with 5 carbon. Then synthesis of squalene, 30 carbon, and then conversion of a squalene to cholesterol, that is 27 carbon. So this is how your 5 stage of production of cholesterol is done actually. So basically we deal in here in the case of lipids here with the cholesterol we have done with phospholipids, plasmalogens and cardiolipids we have discussed in quite details. Then phosphatidylene serines, how their function is. Then we discuss with the cephalins in this case, lecithins, and on also phosphatic acids. So basically these three different types uh, of your phospholipids have been dealt here, and plasmalogens, cardiolipins, and cholesterols. So that's it. That was very short and crisp for you. Now we are done with the lipids also. Let's go for nucleosides now, nucleotides part. Why is it so slow? Any questions so far, bacha? Any doubts with you? So are we going good so far, everyone? I hope you are not slept with my lectures. Okay, good. Uh, let's start the next part. Before we start with the virtual labs, I want to finish with the lectures today because they will make more sense then. Otherwise, we can keep continuing like this. Will not make sense further. So proteins we did already. So I will switch this off. And I shared with you proteins also, right? The message. Yes, protein has been shared. So nucleotides. So we have discussed already. You are all smart students. You know much about this, and so this is the nitrogenous bases. So they are the planar and aromatic and heterocyclic, derived from purines or pyrimidines. So nitrogenous bases are unprimed. So here you can see this is your purine and this is your pyrimidine, right? And in the purine you have N here, and in the pentagon case, by they are being repeated 4 times like this, and there is a disulfide. So this is a benzene ring. So this is how your purine element is made, and you count from here: 1 2 3 4 5 6, then 7 8 9. And on the right hand side, pyrimidine goes from here down: 1 2 3 4 5 6, like this. These are your pyrimidines, purines and pyrimidines. So purines could be of two type, that is your adenine, and pyrimidines adenine and guanine, and pyrimidines could be of three types: cytosine, thymine, uracil in the case of mRNA. So instead of mm, we discuss that. So adenine structure is just as having a an amine group attached to it on the first carbon, right? And then on the other case, in the guanine, you have a double bond oxygen attached to here, and also amine group attached to here. The rest is all same. So that is the main difference between your adenine and guanine. Whereas your cytosine has a double bond O here and a amine group to be attached on top, and it's a single benzene ring, not double. So it's also here double oxygen bond attached to here and methyl group attached here, and like this. Then also here NH attached here, NH attached here, and two oxygen group. So we don't have that is the difference between your thymine and uracil. Yeah, so you have CH3 here attached, and in this case it's not. And in the case of mRNA, instead of T you have U. So A is attached to U in the mRNA instead of attaching to T, and G is attached to the CY triple bond. So that's how the main difference is. So your sugar is a five carbon sugar. Numbering of sugar is primed here in this case. From here we start: 1 prime end, 2, 3, 4, and 5 prime end, and it's there. And there could be two types of sugar. In the DNA it's a deoxyribose sugar, and in the RNA it's a ribose sugar. So this is the ribose sugar in the RNA, with you can see OH group attached here at the second carbon, whereas in the DNA you have a deoxyribose group which lacks OH group at the second carbon. So at the lacks two OH group at the second carbon end. So that's the main difference between sugars of two ribose and DNA and RNA. And what is your nucleosides? It results from linking one of the sugars with the purines or pyrimidines base through N-glycosidic linkage. So with the help of N-glycosidic linkage, for case in the purines bond to carbon 1 of the sugar at the N9 atom, and in pyrimidines carbon 1 carbon of the sugar to the N1 carbon atom. So like this. So this is your sugar, this is your carbon 1, and this is so as we said, in the N9 for the purines and at N1 for the pyrimidines. So this is N9 actually. So this is for the pyrimidines, so it's attached there. In the purines, it will be N1 actually. So that's how the nucleosides are attached to the sugar of your concerned partner. So in this case, adenine is attached to the sugar, so it's become adenosine. So with sugar plus your base pair, with the help of the bond that is called is here is an glucosidic linkage bond. The bond that is being created between the two is known as an glycosidic linkage bond. And now the phosphate groups has a mono, di or triphosphates group could be happen, and phosphates can be bonded to either carbon 3 or carbon 5 atom of the sugar. And nucleotides are made up of results from linking one or more phosphates with the nucleosides onto the 5' end of the molecule through the esterification. So here you can see adenosine monophosphate is there, and it is being attached to the phosphate group, the sugar group, and this is your adenosine group, and this is single phosphate group. So this is called as AMP because monophosphate is attached to it. And here we have OH group attached here. So this is AMP because it's become a ribonucleotide. But in the other case, if there is no attachment of this, then it's a deoxythymidine monophosphate because we have removal of the second carbon deoxy. So this is a deoxyribonucleotide. This will be present in RNA and this will be present in your DNA. So these slides that I'm showing you, they might be you might have read them quite number of times or before, but my reason to explain you again and again in so detailed manner because sometimes we forget them and forget how things are attached and how things are attached another way. So just to keep your memory fresh, these lectures are made to keep you remember them forever with you. And please write down these things in your assignments also, so that either do it with the help of your laptop or do it with your hands. I'm open to both of your courage. So just do it and remember these things with you. So nucleotides are the RNA. Ribonucleic acid is a polymer of ribonucleotides, whereas the DNA is a polymer of the deoxyribonucleotides. So both deoxy and ribonucleotides contains adenine, guanine, and cytosine, whereas the ribonucleotides contain uracil, as I discussed before, and deoxyribonucleotides contains thiamine instead of uracil. So that's the main difference between U and T. And monomer from the nucleic acid polymers are made in the nucleotides. And further, nucleosides and triphosphates are important energy carriers in them in their case, and important components of coenzymes are FAD, NAD plus, and coenzyme A. So they are the quite important ones, enzymes for them. So how do we name them? These nucleosides and nucleotides. So in nucleosides, purine nucleosides end in sine at the end, and that's why their name goes like adenosine, guanosine. Whereas the pyrimidine nucleosides end in dine, so we name them as thymidine, cytidine, uridine. So that's the main difference between purines and pyrimidine, ending with sine and dine, cytidine, uridine, whereas the other ones are uridine and guanosine. And further, nucleotide starts with the nucleoside name from above. Add mono, di, or triphosphate, adenosine, monophosphate, cytidine, triphosphate, deoxythymidine, diphosphate. So they are all being arranged in that case. So that's it from the nucleotides perspective. Uh, so I will stop it here. Take another. You can also take 5 minutes break now. So we have been going through for 1 hour continuously. So we see us in 5 minutes. Oh, I cannot close it for some strange reason. Just a second. Yeah. So sir, yeah, I'm pressing B10. Okay. So students, I see you in 5 minutes. Take a break, chill pill. Then we continue with other things. Thank you. So welcome back class, welcome back. How are you? So let's start. So Sneha asked me about pyridine. Uh, Let me just recall. Hmm. So pyridine is just basically a heterocyclic compound which just has like a benzene ring, like C5H5N, and it is related to benzene part. It's something. It's maybe required in the what you say, in the degradation or biosynthesis of pyrimidines or biosynthesis of acids. So maybe it's required in that part, but not in the in our case. In the case of DNA, might be not so playing big role. So okay, let me check how's our live recording going on. It's going slow, but it's going on. Anyhow, so students, let's continue the other part of your nucleic acids biosynthesis. So, uh, this is about now we're going to talk about pyrimidine biosynthesis, something a biochemistry about the pyrimidine biosynthesis. Furthermore, uh, so these are the following functions of nucleotides. That is, they are the building blocks of the nucleic acids, of course, for DNA and RNA, and they involve in the energy storage, muscles contraction, active transport, maintenance of ion gradient. Further, they are activated intermediates in the biosynthesis, components of coenzymes, metabolic regulators, secondary messengers like CMP, CGMP, phosphate donors in the signal transductions, regulations of or utilization. So they are the various. They have various functions of our nucleotides. So our nucleotide is made up of, as we have discussed already, of your phosphate group, your nucleoside group, including your pentose sugar and purine or pyrimidine base, which are attached by the beta glycosidic bond. And this could be either your nucleotide could be either DNA or RNA based on the ribose sugar or deoxyribose sugar. So this is how everything is arranged: phosphate group, nucleoside group, and this is whole together is known as your nucleotide, including your phosphate plus nucleoside group. So these are, we have discussed various nucleobase structures. Purines, we have adenine, and in pyrimidines we have cytosine, thymine, uracil. There's also hypoxanthine and xanthine in guanine also. More than that, if somebody is interested, if there are for the nucleotide biosynthesis, there are mainly two major routes. That is salvage pathway and de novo pathway. The salvage pathway includes your activated ribose, PRPP, which are required for the activation of your nucleotide plus base pairs, and which give rise to various D-nucleotide triphosphates. Whereas the de novo pathway includes your activated ribose for sure, PRPPs, plus amino acids, plus ATP, plus carbon dioxides. And these are they are giving you nucleotides afterwards, and with including DNTPs. Further, synthesis of pyrimidines. In the synthesis of pyrimidines, you can see you have a bicarbonate here, yeah, HCO3 negative. and then you add glycine with 2 ATPs, giving rise to glutamate plus 2 ATP plus phosphate, giving rise to carbomyl phosphate. Yeah, and this carbamyl phosphate will further give rise to aspartate, giving you two molecules out of phosphate out of it, giving rise to carbamyl aspartate. So carbamyl aspartate will again remove three molecules of water, making you dehydroorotate. And this deorotate with the help of NAD plus give rise to NADH plus H plus, giving you orotate. And this orotate giving rise to orthidine monophosphate, giving you orthodiene monophosphate, that is OMP. And this OMP further give rise to you removal of carbon dioxide, uridine monophosphate, and then further cytidine monophosphate by the addition of glycine ATP and glutamate and ADP plus phosphate group. So this is how your synthesis of pyrimidines reactions happening like this. Starting for bicarbonate, you have your at the end UMP, uridine monophosphate, and cytidine monophosphate. And how, if we have studied the biosynthesis, but how they are degraded then, which is as follows. With the CMP, cytidine monophosphate, first it makes cytidine by removing of the phosphate group. Then water, with the help of water, amino acids groups are made. Then it made into uridine. Then with the uridine, also from UMP, if you remove the one phosphate group, uridine monophosphide, you have your uridine. From uridine, it take one phosphate group to make ribose 1P to make a uracil. And with the ribose 1P, with the uracil being made, it give rise to dihydrouracil in the presence of NADP plus. An addition of water will make a uridopropanate, and hence then further beta-alanine and malanosimildehyde, and further acetyl coenzyme at the end. So this is how you can degrade your pyrimidines also, like this reaction. So what are the defects in the clinical diseases of these pyrimidine biosynthesis? So defects are orotic aciduria. So that is symptoms such as anemia, growth retardation, orotic acid excretions. They are being used in there. And the causes that are happening are a defect in the phosphorybosyl transferase or orotin decarboxylase. So these are the causes and these are the symptoms of it. So here you can see in the reaction here an orotate and PRPP, that is your 5-phosphoribosyl and 1 pyrophosphatate. They comes together, they make a reaction together, and hence PPI is removed out of them, which is a reversible reaction leading you to the orotidylate and uridylate. So giving you these two structures later on. And you can see treatment patients who has these diseases with the orotate aciduria, they are given with the uridine, and this uridine is producing UMP, then further UDP and UTP. And this UTP that is being produced, this inhibits carbamyl phosphate synthesis too, and preventing the biosynthesis and accumulation of orotic acid. So UTP inhibits the carbamyl phosphate synthesis too, preventing the biosynthesis and accumulation of orotic acid. So that was about defects in that, and how it can prevent this biosynthesis of accumulation of orotic acid. So by this reaction, you can see here is glutamine is producing with the bicarbonate and ATP with the help of PRP and carbamyl phosphate synthetase. These enzymes, we have a carbamyl phosphate, and carbamyl phosphate producing aspartate, carbamyl aspartate giving you OMP, then UMP, then UDP, diphosphate, monophosphate, triphosphate, then cytidine triphosphate. But UTP is going back, inhibiting this reaction by not letting produce our further these reactions and inhibiting the production of orotate also. That's how these reactions are done. So summary that we have discussed so far is that pyrimidines are synthesized by the de novo and salvage pathways, and pyrimidine ring is synthesized from the pre-assembled ingredients and then attached to the ribose. And also pyrimidine biosynthesis is tightly regulated via feedback inhibition, that is CTP, and synthetase, carbonyl phosphate synthetase, aspartate transcribable amase, and transcriptional regulation. The mammalian enzymes are multifunctional and form multi-enzyme complexes to increase the efficiency. And drug inhibitors of pyrimidine biosynthesis are under development as potential antimicrobial and anti-cancer agents are being produced in this case. So these are the references for further. So we are done for today from the presentations point of view. Now we will start in the later part of our reactions uh, some virtual labs. So before we start virtual lab, we have been through about DNA and RNA and proteins. So can anyone tell me what is central dogma is? What is central dogma? In the chat, please, could you just tell me what is central dogma is? Anyone? Yes. Flow of information from DNA to mRNA to protein, that is known as your central dogma. And how is it done? Via translation first, then transcription, right? First we have a translation, then we have a transcription, isn't it? Yes, transcription first, then translation first. Yes, that's true. So I was trying to confuse you. Uh, so based on that, how from DNA your mRNA is produced, then from mRNA various transcription factors are playing role in that case. There is a one small video about transcription and translation which will be good to produce here in our case so that you can understand about the whole part of nucleic acids, this DNA and RNA that we have discussed now, which will make some sense to this to its application, how this DNA and nucleotide and nucleosides are playing role in further actions. So I will play this video now. It's a quite long video, so it's quite interactive also, and you will learn a lot from this. So let me zoom a bit. It's too much zoom, is it? No, it's okay. So
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
it's asking us to choose a gene. Let's choose this gene.
Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases. When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try. When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, a proteasome, will now chop up other proteins for recycling. Proteins get sent to the proteasome when they have finished doing their jobs, when they are damaged, or when the cell determines that they have come from an outside invader, such as a virus. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
that was quite uh, nice video about transcription and translation about central dogma, how your mRNA produced from DNA, then it's going out of your nucleus, then from there how ribosomal RNA came, and with the help of transfer RNA, with the different anticodones, they're producing your amino acids, and there's a long chain of amino acids are being produced, which are able to produce your proteins, and they're forming into their post-translation modifications and forming their structures and started to functions for the things. That's a summary of all this, and I will share this with you. So that we do not goes off the topic. So guys, I think we are done for the lectures today. That's I want to teach you today all in all. Now in the rest of the period, I want to take you to the field of how you can isolate DNA, and after isolation of DNA, if time survives, we go for PCR. Like if we have isolated the DNA, then what we have to do is increase its production of DNA, because what we get DNA in the amount is get very low in amount. So even when there's a COVID tests are being done, so they do RT-PCR, reverse transcriptional path PCR, to test your COVID test. So we will do with that pulse that part as a virtual lab. We will understand that thing. So to do so, let's start for the next part of your workshop today. So DNA extraction. So let me zoom in. So let's start how you can extract DNA from human body. So in this experiment, you need to isolate some DNA from human test subject. And why, you may ask, why do we need human DNA? Let's check. So let's go for genetic testing, body identification, or for the analysis of forensic science. So scientists are doing to check this DNA for variety of reasons. That is to check whether there's any genetic testing has to be done, body identification has to be checked, or analysis of forensic evidence has to be done. So for the following reasons, we do the DNA isolation. So DNA extraction is typically the first step in the longer laboratory process, and DNA extraction is an important part of that process because DNA is first needs to be purified away from the proteins and other cellular contaminants. So we need cells that that has to be purified away from the proteins and other cellular contaminants. So we need cells because that's where the DNA is inside of it, and we have to check the nucleus about 2 meters of DNA. So it's a quite a long process to do so. So the cell, you have a nucleus. So inside the nucleus you have chromosome, and inside the chromosome you have histones, and around the histones your DNA is coiled. And this is your double helix DNA with various base pairs, amino acid base pairs. So first we need to collect some cells from the test subjects. To have this DNA from the cell, we need to initiate this thing. The skin on the inside of our mouth loses thousands of cells every day, and these are the cells that we are here after. So let's start the experiments by doing four different steps. First, we collect the cheek cells, then we burst cells open to release DNA, then we separate DNA from proteins and debris, then we isolate concentrated DNA. So by these four methods, we can have our DNA be isolated completely. The first thing is first to let's get started to have a purified DNA from our cheek cells. From the cheek cells we are getting, let's start. So here are the materials and equipments you will need to purify DNA from the cheek cells. So you need water bath, centrifuge, micropipetter, buccal swab, sample tubes, lysis solution, concentrated salt solution, resuspension buffer, ethanol, and also isopropyl alcohol. So first we drag the swab over the inside of the test subject to collect the sample. To collect our sample, we take the buccal swab and put in the mouth. So now you can see that in the swab we have our cheek cells being collected on the swab. Now we will put this in the Eppendorf so that covered with 100 of tiny cheek cells. Inside cheek cells is a nucleus, and inside the nucleus we have our DNA present. So we need these cheeks to be present in the Eppendorf tube now. We cut it and put in the Eppendorf tube and we add lysis solution to it. So this lysis solution, what it will do, it will remove all the cell part, it will open up it and separate from this other cell debris, and our DNA will come out of it because of the help of this. So we put this on the warm water bath at a particular temperature so that the reaction could be start at that temperature. So your cell wall is broken, your nucleus is broken, your chromosomes are broken away, and then from the chromosomes your histones that were present, they were also broken. So what happened actually in this case, the lysis solutions that we have, it has two important ingredients. That is, there is a detergent, an enzyme called protein kinase K. What detergent is doing, it's disrupting the cell membrane, nuclear envelope, and causing the cells to burst open to release their DNA. So it is helping to remove the DNA out of the cells by breaking up the cell membrane and nuclear envelope and causing the burst cells to be open. Whereas your DNA is still up wrapped with the tightly around these histones, and protein kinase cut apart the histones to free the DNA. So proteinase K will cut these histones from the free DNA. So go next. So we're gonna separate this DNA from proteins from the debris. To do so, we add concentrated salt water into that. So the salt water, it will do, the cells will have stayed in the warm water for both long enough for the DNA to be freed from the cells. So we have to remove this swab from the tube and add some concentrated salt solution to your tube. So let's add. So what will happening inside now, because of the salt solution, DNA will come together, will separate like together, and all the cell debris clumps together. So this salt causes proteins and other cell debris to clump together. So that's what salt is doing. and DNA is separated from it. And to separate the cell debris from the protein, what we will do, we put this into the centrifuge, and at the bottom we will have the cell debris and the top we will have the DNA. And this is to keep balance between two. We have to put another tube on the other side. Otherwise, if you run this machine, the machine could not will not work. It will make bad sound. So you have to keep it balance in front of it. So centrifugation machine is working on now. There you go. So you have proteins debris to be down, and your all cell clumps to be present and up, and hence your DNA remains distributed through the liquid, and you can have your experiments keep going on. So use the micropipetter to carefully remove the top liquid so that we can have our DNA out of it, and place it in the clean tube. And now we're going to isolate the concentrated DNA by adding isopropyl alcohol. So we're going to add this isopropyl alcohol to this slowly. And remember, when you're using this micropipette, don't do it with a flash like zoop zoop. Don't make it flash, because if you do it very fast, things could come outside. So do everything when you're in the wet lab. This is wet lab working. Whenever you're in the wet lab, do it everything slowly and gently, so that, and mix it very well upside down. So like this, very slowly he had put up the things, nothing is coming out, and with the hands gently move up and down. And what you will see, by moving up and down this gently, DNA is clocked together, and hence you have a DNA. Because why, by inverting in the isopropyl alcohol this was happening? This was happening because DNA is not soluble in isopropyl alcohol. It does not dissolve in isopropyl alcohol. So it's come out of the solution. So why does not DNA dissolve in isopropyl alcohol? Why is it so? So that's why we are seeing clump of DNA with our naked eye in this case. Why isopropyl alcohol? Why our DNA is not dissolvable in that? So I will discuss this protocol. I have a protocol for that. So you can go through that protocol, and you can calculate the values for the same. So don't worry. So why isopropyl alcohol is used in that case to remove the DNA? Because it's a polar reagent. So in that case, our DNA does not dissolve, and hence we see these clumps in that. So to remove that clumps, what we will do, we will centrifuge this, and get the at the bottom of our DNA will be at the bottom, and the rest will be isopropyl alcohol, and we can keep it safe for a very long number of days, and use it for as ever whenever we have to use it. So we close it. You isolate this. So now you have your DNA at the bottom. So once the liquid is removed, DNA is allowed to dry, and you can redissolve in the solution of your choice. You can store it in the freezer for many years, or you can move it on your next experiments. You just have extracted DNA. So you can do this in the kitchen items or anything, whichever you feel like, and goes like this. Uh, so that's it. I think that we will not go for further. There is error for this. So I will share this exciting workshop with you, DNA extraction workshop, and discuss with you now the protocol, if I have one. So this is for the next day. Uh, this is where I was teaching at the Fatehgarh Sahib in Mata Gujari College as assistant professor of biotechnology to the agriculture students. So I was teaching principle of plant biotechnology. So there I had this chance to do these experiments. So this part of it. So after doing it, uh, how what is DNA and how does it work? This is another video for the same. Start ahead.
Stated Clearly presents What is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function.